a visiting fellow at the British Academy, but what he's probably most known for are his books, one of which is Kluge, which I think was a very, very popular book, The Haphazard Construction of the Human Mind, a more recent book, Guitar Zero, The New Musician and the Science of Learning, and I'm going to plug his brand new book, The Future of the Brain, edited by Gary, essays by the world's leading neuroscientists, and there's a little something in here for the whole family to enjoy. But it's coming out in November, so I highly recommend that you read it. And what he does is he makes science accessible, very readable, and enjoyable. And unlike a lot of people who do these sorts of things, he's actually quite accurate in his information, which is good. And you've probably heard him a lot this week. He's contributed a lot to our conversations on evolution and development. So I think that's all I'm going to say, Gary. Is that okay?
Sure.
Gary Marcus, thank you for coming.
Thanks very much. I'm not going to try that hard to make today's talk either accessible or entertaining or any of that kind of stuff, but you know, we'll see how it goes. Instead, based on watching the other talks take like 3 hours and including costume changes, I decided that you guys wanted a little bit of detail, so stop me if there's too much or too little or whatever. The evolution of language raises a problem. This cartoon depicts one of those problems that the evolution. But that's not the one I'm going to talk about today. The basic problem that I'm going to talk about is that a lot has happened relatively recently. I tried to squeeze Leah's wonderful chart about what happened over the last 400 million years down into a little bullet point in the slide. So there it is. A lot has happened relatively recently. So she suggested that language might have emerged 700,000 years ago. There are other people that suggest that it happened 50,000 years ago. Whatever it is, it's a blink in the eye of the cosmic calendar. And not that much happened genetically in the sense that we obviously, by inference, share a lot of DNA with our last common ancestor. Chimpanzees aren't our last common ancestor, but we share more than 95% of our DNA with them. Our brains, to first approximation, don't look that different from chimpanzee brains, except with respect to their size. There are no known human unique cortical areas, last I checked, no known human unique cell types. Sometimes people get excited about things, but like spindle cells, and they say, oh, these are only in primates, and then eventually it turns out that that's not true. And there's no cell type that I know that's uniquely associated to people. But so that sort of leaves size, but size isn't everything. Pygmies and toddlers can talk, and even the largest brain chimps can't. I suspect that size is a contributor, but it's not the only explanation for why one of these guys manages to be able to talk and the other one not. My best guess, I know it's a trick question, my best guess is that the standard tools, which Leah also summarized nicely, and I decided I would get this one up on a slide, the standard tools are probably part of what's going on. And by standard tools, I mean that we can look with the kind of coarse grain tools that we have now, and we can say this area is bigger, or it makes more connections to this other area, and so forth. My guess is that those tools aren't going to be sufficient to explain what's special about the human brain. I don't want to say that they're not necessary. I'm sure all of those things are going to contribute. One of my first questions at the conference was about what I called letter H, which is connections within cortical areas. And in a way, that's what I'm going to be talking about today. Not in as much detail as I wish that I could. But, so I think these are probably necessary mechanisms, but I think that ultimately it's going to come down to microcircuitry, basically, in particular cortical areas that are subtly different. So I wrote this book, which I'd love it if you guys would read. It was not my most popular book. It was my first attempt to write a book that was accessible to the general public. I think the New York Times wrote a very short review saying it's great when it doesn't get bogged down in molecular biology or something like that. So you guys might appreciate it even if the Times didn't. I think that it tries to address many of the issues that folks in this room try to address. And I mean that as the sort of union of a set of interests here. So that book was about the relation between developmental biology and developmental psychology. That's really where my training originally was in developmental psycholinguistics, trying to understand how children acquire language. So I wrote what's at least technically called a trade book about trying to put all this together, put all the molecular biology together with developmental psychology. What I argued there, this is a 2004 book, but I still stand basically by the overall argument, is that the most important factor in explaining the difference between humans and chimps is probably cortical area internal microcircuitry. part of the premise of the book is there's probably not enough room in the genome for language to be specified from scratch. I used to go around showing a headline from the San Francisco Chronicle when the genome was sequenced. It said, humans survive on fewer genes than thought. So, you know, Gilbert made up this number, there are 100,000 genes. I should have left that, and I'll show you later. Gilbert made up this number that there were 100,000 genes. He did it in the back of an envelope for a textbook, and it spread everywhere. And then it turned out there were 30,000, which somebody repeated yesterday. I think the actual number is down to 20,000, but you know, what's 10,000 genes between friends? So the point is there's not a lot of bits in there to be specifying, I think, a language faculty from scratch, because all those bits of information also have to specify how you grow your kidneys and how you grow your lungs and so forth and so on.
You're going to tell us more about these microcircuits.
Well, I'm going to...
Pester you on that now.
You can start to pester me now. I'll tell you now, as I will on a slide in a minute, that I don't have an answer. I have a shift in direction that I'm going to try to pedal.
But just for clarification, what's micro in this scale? It will be at the level of, let's say, a few hundred micrometers, or?
At the level of connectivity between individual neurons, not cortical areas, but at the level of individual neurons. I'll even hold open the possibility that some of the microcircuitry could be inside a neuron. So it could be that single neurons do really interesting computations that are a lot more interesting than sigmoids. So it could be very micro. And I think that one way, at a very abstract level of thinking about the whole field, is the coarse-grained tools that we have, and they're not that coarse-grained, but haven't revealed the differences that make the explanation. And so either the explanation has something to do, say, purely with input, which some people would try to explain, or there's something below the level of detail that we understand that's really important. So part of what I was trying to do in this book and also include was to explore the implications of Monod's idea that evolution is a tinkerer, which is really just Darwin's idea of descent with modification. That new circuits are built on top of old circuits, or any biology is built on top of old. So you don't just start from scratch building a custom solution to a particular problem. You build something that works with what's already there. So old circuits get tweaked to do new things. It's not like the language faculty is custom designed to do language and half of the genome does language and nothing else. Or at least it's probably not that way, is my guess. It's probably more like there's a lot of circuitry in there already. There's some changes to that circuitry that give you something new, but there's a very strong starting point. One gut instinct that I have, and that's why this slide is called my bet, not my proof or anything like that, is that a circuit that repurposed memory in a very particular way could be really, really important to giving us human cognition. And that particular way, for those who have done any kind of computer programming at all, is in computer programming you can store different kinds of numbers. You can store integers, you can store strings, and so forth, different kinds of information or other. And then somebody came along and said, well, we could have an ordered way of storing information such that, for example, you could have lists of words like sentences. One popular language, maybe the place where it was invented, is the computer programming language Lisp, which is the pioneering language for artificial intelligence. John McCarthy invented this language, and you can put things together so that when you store things in memory, it's not just a bunch of isolated elements, but there's a structure to them. That structure is also closely related to the tree structure that you see in the folders or directories of your computer and so forth. If I had to guess one thing that really made our cognition different from other creatures, it might be that we have this way of storing information that allows us to store information with structure, so that we can, for example, represent the tree structure of a sentence. Have you guys all seen a linguistic tree structure? I should have had one on a slide here. So it's like an upside-down tree. It says a sentence goes to a noun phrase and a verb phrase, and a noun phrase is made up of, let's say, a determiner and a noun, and a verb phrase is a verb and a noun phrase, and so forth. So you have this branching structure. Well, that's critical for language. It might be critical for doing abstract planning, for doing a lot of abstract representation. That's a representational format. It's not an algorithm. It's just a representational format to even be able to store something like this, pull back information from it, access it. Could be that something like that would have a dramatic change, and that itself could be a tinkering on something that came before.
But Gary, I could argue that if I look at the generation of behavior, I might find a similar kind of embedding, right? That, for instance, has to go from A to B, but in order to go from A to B, you have also the sub-actions it must perform, for which I have, again, control my skeletal muscle system in a very structured way, right? So that on the tree structure, I might already find that it's that has to perform action. Or in the
You might.
right? So
And I think there's actually a couple options there. One option is that in other creatures, the ability to do that is actually limited to, say, response chaining. I don't know. Dorothy actually has some data that I think are somewhat on point here, but I don't know how that literature really shakes out. But it's possible that you get a long way with just response chaining alone without having the ability to really represent fully abstract structures. I don't actually see Dorothy here, but there she is. So she's got some interesting data that maybe she'll talk about tomorrow that's relevant. So that's one possibility. Another possibility is there is the ability to represent, let's say, recursive tree structure in the context of motor in other creatures, but we've somehow freed that ability to use it in a much broader way. And I think that that's a perfectly reasonable possibility, too. That also would be a kind of tweaking relative to a lot of machinery that came before. And I would also point to we need to understand something about the microcircuitry. I guess that latter possibility could turn out to be connectivity between areas or something like that. So, I mean, I think that's on the table, too. More broadly, and you can continue to ask questions, but more broadly, the way I think about it is kind of something old and something new. So rather than trying to understand a language module, which is kind of the way I was trained, I was a student of Pinker's, who wrote this book, The Language Instinct, which you've probably heard of, and we both were in the sort of sphere of Chomsky, and Chomsky talked a lot about a language module. And the idea of the language module is there was some special purpose device in the human that allowed them to acquire language. And Chomsky often used this phrase of autonomy of syntax. The idea was that the grammar of language was separate from the rest of cognition. And I believed that when I was in graduate school, but I don't really believe it anymore. I think that a better way to think about it is that language is kind of made up of overlapping circles of lots of other abilities. There may be a little bit that's unique to humans, probably is, almost certainly is, but the machinery that gives us language is overlapping. Whoops, I'll try to do the animation again, sort of backwards. Overlaps with the machinery for motor control, for cognition, and so forth. So like Paul's question just asked, I think motor control is a big contributor to language. So machinery that allowed us to put together any bit of sequencing in order to do action over time, that's going to be important, for example, in language. So there was something funny about the argument in retrospect, which is this, that if you look at the correlation, I'll take a step back. One of the pieces of evidence for the autonomy of language was the idea that there are some special populations where people have intact language but impaired cognition and the other way around. So people with Williams syndrome have almost perfect language, actually it was exaggerated, but have pretty good language and poor cognitive skills, and then people with specific language impairment have pretty good language skills and impaired, sorry, I said that backwards, have pretty good language and impaired cognition. And so people went around talking about these examples as a kind of double dissociation. But if you looked across the population, verbal skills and say, math skills are actually correlated. So SAT math and SAT verbal, for example, are well correlated. And that doesn't really fall out of the modular kind of view, where language is a separate module. I think that the modularity hypothesis can still be rescued, but I don't think that the modules are language. I think the modules are computational things to do particular kinds of, say, things like sequencing or abstraction and so forth. Okay, so anyway, this is not a modularity position in the traditional sense. This is a position that says that language is a tinkering of old parts, probably with some new parts, so hence the something old, something new. Okay, but then we come directly to Paul's first question and my honest answer, which is I have no idea what the circuits are. I wish that I did. But still, even though I don't know what they are, I'd like to suggest a shift in direction that I would like to think might help get us there a little bit faster. So the rest of this talk, the first part kind of consolidates a lot of my own work from many years, and I've left out many details and could talk about it for hours. The second and third parts are in collaboration with Adam Marblestone, who just finished his PhD with George Church and is now a postdoc in Ed Boyden's lab, and Tom Dean, who's directing some of Google's neuroscience efforts. So neuroscientists often write, and I don't know what they really believe, but they often write as if they were on a quest for a single canonical cortical computation. Here's an old version of this. The neocortex can be understood as a cooperative network that acts as a nonlinear spatiotemporal filter with adaptive properties and that transfers signal flow. It is assumed that these filter properties are identical for all neocortical areas. The functional role of a circumscribed cortical area depends exclusively on its position within a certain functional circuit and is defined by it. Now, this is a pretty old quote at this point, but I think it still represents the thrust of a lot of computational neuroscience. I don't think it would be out of place in a modern discussion. I think we have better tools for articulating theories like this now, but I think that this is still a pretty common view. And notice that it leaves most of the difference between different cortical areas, hence, say, the differences between language and vision, to functional input. So the idea is you've got one kind of circuit, it's just copied over and over again. That six-layered sheet that John was talking about obviously would play a role in whatever that canonical cortical computation is. And on this view, and I'm not saying everybody believes it, but I do see traces of it a lot in computational neuroscience. The idea is you've got this one circuit. If we could just crack this one circuit, we'd be done. And there's a way, actually, in which I think Markram's what's it called, the Human Brain Project, is dependent on something like this being true, because what they're trying to do is define basically a canonical circuit, build a very detailed model of that, and they're hoping that that will generalize across the brain. So I think there's a lot of money at stake here too. I think it's a real idea that has real instantiation. And here's some other quotes. Another quote from about the same era. All parts of the neocortex might operate based on a common principle. That's a paper by Mountcastle that's cited all the time. And then there are various other versions. The one in the middle, I wanted to highlight this part about only a few computational principles, that's all we need, and then the variability of the input signals yields functional specialization. So that's one theory. When I say that I don't think size is everything, you could say, if you came from a particular school of computational neuroscience, it's not just size, it's that the input is different. So the input to audition and the input to vision are different, and particular language has a kind of structure, but it's all one kind of circuit type that's cranking away. We have more of that than the chimp, and that's why we are able to acquire language and they aren't, but it's all basically the same kind of computational stuff.
But in that paper with Rita Weiss and Peter Koenig, we did show that based on that idea, you can regenerate a whole cortical hierarchy that is physiologically possible in terms of the physiological signatures that it generates. Right?
So hold that thought. I have an answer to it,
Okay, cool.
which you may or may not find satisfactory, but it's a long answer, and it's sort of the next third of the talk. Borrowing 15 minutes. I see your 15 minutes and double it.
Yeah,
Question.
What is your definition of a column, or is it completely meaningless?
I don't have a definition. I kind of feel like that's over my pay grade. I see my mission as unifying a whole wide range of things from cognition to molecular biology, but I'm a generalist. I'm not like, I would yield to many people in this room. I'd probably start with John, but many people in this room are going to have a clearer idea of that. And I mean, I know that there are arguments about that. I don't have a stake in them. I'm not even sure the cortical column is the right unit of analysis. I mean, this is, I'm not committed to it.
Column, we use all sorts of definitions, all sorts of sizes.
Yeah, I'm aware.
Basically, nobody argues that there is a radial arrangement in the cortex, and you have different modularity. But when you start comparing 30 micron thick microcolumns with 300 micron columns, then there is a bit of a problem, I think.
I mean, I think where I'm going is going to suffer from the same problem. I agree that there is such a problem. I mean, we don't really know the basic unit of analysis, right? I completely take that for granted, and that's going to be problematic for what I'm suggesting as an alternative course too. And I don't really have an answer to it. I think it's an important question, though. So before I try to argue against the canonical microcircuit view, I want to say why I think a lot of people have actually believed it. And I partly want to do that because I've given this talk, the first few times I gave this talk, people said nobody really believes that. And it's certainly in the literature, and people talk about it. Maybe they don't really believe it. Sometimes I get the line that computational neuroscientists might believe it, practicing experimental neuroscientists don't. And I don't really know the state of play, but I want to talk about the view, why people might believe it, and why I think they shouldn't. So one reason, and John alluded to this already, I had sort of somewhat similar slides, is that the cortex is surprisingly uniform between areas and across species. If you had asked me in 1990, when I was a student of Pinker's studying language, didn't know much about neuroscience, and we didn't have a lot of the techniques that we had available now, and I was basically ignorant, I would have said, well, I expect that Broca's area just looks a lot different from occipital cortex. And there's no reason now to really believe that that's true at the grain levels that we can look. It is, I think, a surprising fact, given the diversity of cognitive function, that the cortex is relatively uniform. So if you started a priori from the psychological data and maybe some lesion data or something like that that told you that different areas do different things, you might think, in accordance with the fact that different areas do different things, that anatomically they would look really different. And I think it's a basic important fact that has to be explained, that they do look, at least superficially, fairly similar. That doesn't mean that at the finest grain level that they're all identical. And I think sometimes people wander into the latter inference that they shouldn't. But there is a surprising amount of uniformity between areas and across species and so forth. I think, you know, we all expected that chimpanzee Broca's area would look, or homologue to Broca's area would look really different from human Broca's area, and that doesn't really seem to be true. Sorry.
Overstating the case of uniformity. I mean,
I'm going to come back to that. I'm going to come back to it.
I mean, the original staining, which wasn't some, you know, massively sophisticated thing, defined the areas by their side architecture.
This is an important observation, yes. I know it. I might even have it on a slide. I mean, yeah, exactly. The Brodmann areas are a counterexample to this. And yet, I still hear neuroscientists go around all the time saying, look, there's a uniform six-layered cortex, and I'm going to go look for my canonical computation. And I see it in neuroscience. I see it in psychology. There was this famous book, Rethinking Innateness, by Jeff Elman and Liz Bates, that was one of the most influential books in my field for a while. And this was a central part of their argument, was the uniformity. It's true. I would petition the audience to send me a really good review that really goes through the systematic differences. Maybe John has actually written one. I know bits and pieces. The sort of canonical statement is actually something like, there's relatively uniform, but what is it, layer 4 of motor cortex is agranular or something. Like, people will give a sentence of, but there are some counterexamples. But I think the modal way in which they present this, despite the obvious facts about Brodmann's areas, it's mostly pretty similar, and there are some exceptions here and there. I have not actually seen a really good systematic review that tries to balance the two sides of this. The information is out there, but I don't see it stated in a concise and useful way.
Could you just clarify, are you making the argument currently in these slides for these other people, or are you accepting these premises?
No, right now what I'm doing in the next 6 slides or something, going back to here, it's 5 reasons why you should, why one might take the view seriously, the view that I'm then going to argue against. So I'm giving you the motivations now in the next few slides, the kinds of arguments that I hear for why people do think, oh, well, we are going to find a canonical microcircuit and all will be revealed. So, like, why is it not completely crazy to do the Markram project of, we're going to get all this data on a somatosensory column and we're going to build a simulation based on that? I mean, why think that that has any face value at all? And the next set of slides is going to be an argument for that. Ultimately, I don't think it's a good argument. I think the Brodmann stuff that you raised is, in fact, exactly why you shouldn't take the first argument seriously. But I wanted to lay out the logic for why one might be inclined in this direction. The second is that ostensible functional differences sometimes can be changed, can be derived by small parametric changes. So there are various papers in the literature for people where people have tried to say, I can get both prefrontal function and motor function or something like that out of a single model where I just tweak a little parameter. And so they say they're just very, very minor variations on a theme. Then the study that I think is most interpreted in contemporary neuroscience, but also one of the most frequently cited in developmental psychology anyway, is Mrganka-Sur's studies of, quote, rewiring that aren't really rewiring studies. So they're always described as if what Sur did is he rewired visual input into auditory cortex. And the problem I have is with the word rewiring. So it's not as if he took a scalpel and did the wiring by hand. What he really did is he removed the target, the usual target for the visual input. And the way I would put it is that input then found the next best target that it could find. So you could think of a dating analogy. It was trying to find the perfect match. It couldn't find the perfect match. So it found something else that happens to carry the same F receptors and ephrin ligands and so forth. And it's organized in a somewhat similar way, and it went over there. But people take these experiments as if they're showing the cortex is basically the same stuff. You take a little from auditory areas, from visual areas. Of course, we know all kinds of plasticity effects where you know, blind people reallocate occipital cortex a little bit and so forth. And people leap from that inference, or from those data, to the idea that cortex is generally interchangeable. And I think what it licenses at most, and I think even this is being charitable, is that there's some interchangeability between some sensory areas. So CERN never did these experiments, and I mentioned this in a comment a couple of days ago, never did these experiments or never published these experiments outside of these particular areas. So there are no published demonstrations of the same kind of interchangeability where you would say, take auditory input and send it to prefrontal cortex or something like that. I asked him about it in an email, and he said, we didn't try that hard, which sounds to me like a couple of postdocs worked for like 6 years and couldn't get it to work.
There are some experiments of people that have connected strange devices to
Strange devices? You
yeah,
said?
strange electronic devices that allow blind people to actually see objects. Like those experiments with this device that they connect on the tongue, and then they use.
They're still all
these
sensory
as-
areas. They're still all sensory areas. I mean, I think those are interesting experiments, and they're certainly telling us something. So you can do it to the tongue or the back and so forth. But I don't think it's implausible to think a priori that a few different areas that are all organized by basically the same topographic principles to work on input might have some degree of interchangeability, but that other areas that work in different ways might not. I've never even seen that with olfaction. When you can get the nose to do that, then I'll be truly impressed. Paul.
Gary, so absence of evidence doesn't mean that there is evidence of absence, right? So in that sense, that the experiment has not been done doesn't mean that this interchangeability concept doesn't hold. It just means it's only been established for primary sensory areas.
I sort of give yes and no. I mean, this is why on that. This is why I asked Sir if it had been done. I mean, it's true. Maybe somebody will figure it out. What I infer is it's at least a lot harder. I'm sure after he had his Nature paper and everybody was interested that he did in fact try and he couldn't get it done. It doesn't mean that somebody else couldn't come along and find some other way to do it, but it's at least harder.
I don't think you can really talk about this without looking at it in a developmental context. So probably at a sufficiently early stage, this sort of
Absolutely.
equipotentiality is there right across the cortex, but then fairly soon it gets something gets established that means you can't be interchangeable. And the remarkable thing about the human brain, I showed it on the first day in a slide, is that we're born with a cortex where the neurons are there, but they're not wired up yet. So the wiring waits until we have experience.
Whoa. I was with you till that last part. I really want to talk about that last sentence before you move on. So so I totally agree about the equipotentiality depending on the stage of development. So it's clear that if you do any kind of explant, transplant experiment on early organisms, it's going to work better than on an adult experiment. But people leap to the inference that experience is then the relevant driver of what's going on. But genetic factors can matter too. So it's not necessarily the case that if you find a lack of plasticity later, it's because experience is the only thing that's relevant there. I mean, really, experience is going to be mediating molecular programs. I mentioned the other day the example that I always liked about putting a presumptive retina cell in the stomach. That's not a thing that we want to describe as learning, right? It's that the presumptive retina cell carries a set of genetic instructions that get activated by a particular environment.
But so I just finished. So I think you're born with a very immature brain, but your sensory systems are working. So it's almost the opposite situation, for instance, with the rat, where it's born, the eyes are closed. You know, the sensory systems aren't working, but it can move around a bit more. But with the human, the sensory systems are working, the motor systems aren't really wired up yet or working very well. And something like language is taking 18 months before it really starts to kick in. So why are we born so immature? It's probably because we need that experience in order to wire up the circuits for something like language.
Well, even there, like the last part seems like a leap to me. I mean, some things I'm not sure that are experience driven, but are postnatal. So I wouldn't say that postnatal necessarily entails experience. I mean, facial hair is very postnatal, but it's not very experience dependent. So I do think experience is obviously critically important for language, but I would caution about attributing all of the organization of language to postnatal experience as opposed to maybe there being important molecular cues. I mean, there's Nim Chimpsky, right? I mean, you can raise a chimp as a human being, it's still not going to acquire language. So there are some important molecular factors too. John?
Hate to distract you even further from your talk.
What was I talking about? Where am I?
But I had a long talk with Mariganka on a train ride once when we were going someplace.
On a train ride or a crane ride?
Train, a real crane.
Crane you wouldn't talk.
But this is where we disagreed, and probably still do. And I don't think that the redirected visual information into auditory cortex ended up subserving vision. And he thought it did. And that is sort of a hard question to answer when you've got hamsters or ferrets or whatever.
And that's why I said at most. Like, I mean, if you look carefully at the data, it's not totally obvious what's going on. So I think the most charitable interpretation. Anyway, I mean, these are motivational arguments. I have a counter argument to give in a moment, and I don't want you to be confused into thinking I believe all of these things. Another argument that you could make, which has come from Markram's group and my friend Sean Hill, is the apparent stereotypy of local connections within the cortex. So they have shown in this very interesting PNAS paper, which I think they're going to follow up shortly, that you can take basically a random selection of somatosensory cells, look at the connectivity at the synapse level, and make pretty good predictions looking, let's say, at one rat to make predictions about how the other rat synapses are going to be organized. And so this seems like an argument. I'll come back to this argument too. I'm also skeptical of this argument, but ostensibly it seems like an argument that connectivity is so stereotyped that you don't really need to know the fine-grained detail. There's just the kind of generic algorithm that is putting together the cortex. I'll come back to that stuff later. Then another argument is a computational one, and Paul alluded to this in was his third or fourth of 7 questions that he's asked so far.
Stop counting.
That was a joke, but you were engaged in your tablet. It was a joke for you. I thought you missed it. Anyway.
No, I got it. I'm making notes, actually. You won't believe it.
How can you ask all the questions and take notes at the same time? Okay, so.
It's my feminine side. Multitask.
All right, I'm going to stop this line of questioning. So there are arguments, some of which have been made by people in this room, which say that you can, for example, derive a hierarchy of features using unsupervised learning algorithms, for example. And I take those arguments to be true. It's not going to be where my argument goes. I'm not going to deny that with unsupervised algorithms you can derive a hierarchy. And in fact, I think there's very good computational work. Paul was at the forefront of it, and deep learning is doing a lot of the same kind of thing nowadays in commercial context. So the fact that you can build something like a Hubel and Wiesel model, basically, with a single generic algorithm is exciting, and it tells you maybe about how at least some piece of the cortex might get wired up. And some people have taken that, the fact that you can get those hierarchies there, to think that, okay, we're on the right track here, and I'm going to try to make a kind of counter-argument.
Here, can I say something?
Sure.
Because we're not trying to build a Hubel and Wiesel model here, right? Hubel and Wiesel will be describing, let's say, in a very primitive way, the organization of a single cortical area. But what you try to do here is, but how do I build a hierarchy of visual processing that would go from, let's say, V1 up to entorhinal cortex? That's the goal here. It's not the Hubel and Wiesel model you try to build.
You may think that you're tarred with a broad brush then in the next section. Let's come back to that point. I mean, I think it's a fair discussion point. But let me lay out my argument, and then it's relevant, though. So, and then another argument, which I guess I'm sort of tipping my hand a little bit, but why say that there's a bunch of circuits rather than just one canonical microcircuit if one would suffice? So, I mean, this is part of Paul's argument is, look, if we can get this hierarchy all the way up to entorhinal cortex with one learning rule or something like that, then why should we, propose 15 different stories for 15 different areas. And I think that's a very reasonable argument. Though, again, I'll have something else to say about it. So there's a lot of people out there, bottom line, looking for canonical microcircuit. And I think there's reasons why they might do that. The canonical microcircuit is, in fact, something like Hubel and Wiesel feature detector. So you've got simple cells that go into complex cells that go into still more complex cells. I assume everybody in the room knows the basic idea. I had one quote saying that this is the greatest single influence on the ways neuroscientists think about the brain during the second half of the 20th century. That's one of those grand statements that sounds very hyperbolic, but it's probably actually true. Probably is the biggest influence in the second half of the century. And it's consistent with a wide range of empirical data. So I'm going to argue momentarily that I think that there's something wrong with thinking about the whole cortex in this way, but I don't think there's anything wrong with thinking about some parts of the cortex in this way. I think there's lots of data to suggest that Hubel and Wiesel certainly were on the right track. There's a reason why the idea has become so influential, which is there's data for it that pop up at every turn. They had their own original data in the 1950s, and now they're wild extrapolations that even they would have been surprised about, like the Jennifer Aniston cell that Christoph Koch and Itzhak Fried and so forth documented, where you have individual cells that our best guess, we don't really know how they're wired up, but our best guess is kind of sit high in this kind of hierarchy responsive to very abstract things like Jennifer Aniston, either the name or a picture of the person. You look like you have a question, Tony. No? Okay. No question. Where's the right arrow key again? Okay. And so the Hubel and Wiesel stuff is the impetus behind a wide range of computational models. I don't have Paul's model here, but here's a sampling. There's the neocognitron model, which I think is actually a direct inspiration of deep learning now, which I have on the right. There's lots and lots of models out there. What's cool about deep learning, which has been in the news a lot, is that instead of having a bunch of knowledge engineers hand design the features in your hierarchical representation, you can actually have a generalized algorithm that sorts them out, and Paul had a different approach to that. Probably the most famous instantiation of this, because it appeared on the front page of the New York Times, is a, quote, cat detector that Google built that's basically doing hierarchical feature recognition and taught itself to recognize cats by watching a lot of YouTube videos. What they don't tell you is a lot of YouTube videos are about cats. They did better on the more frequent stimuli than less frequent stimuli, and people love videos of their cats. But anyway, even with the full resources of Google, the system wasn't on par with human performance. It might be part of a solution to explaining how vision works, but obviously not the full solution. I don't think hierarchies of feature detectors are wrong, but I don't think they're sufficient either for artificial intelligence, which is something we're not really talking about today, or for neuroscience. So now I'll give some reasons, some of which we've already raised in discussion, for why I'm skeptical of the canonical circuit view, despite the appeal. One is there's no satisfactory account of what the canonical circuit might be, if there is one. There's certainly no consensus. People have been trying to do this for a long time. I like to quote Bono in this regard at the bottom. If it's out there, we still haven't found it. All right. Second, I think that the canonical circuit. Oh.
Look, we cannot. Where's the microphone, Tony? Until she uses the microphone. Okay, so look, people like indeed, Rodney Douglas, Kevin Martin, Henry Kennedy, who will be here also later tomorrow, have done a lot of very detailed anatomical studies to try to reveal, let's say, basic wiring schemes of cortical circuits, right? Mm-hmm. That kind of data we cannot just swipe away like that, right?
Right, but there are two possibilities here. One is that all that data is relevant to a subset of cortical computations, and the other is there is one cortical computation, and all of that data is trying to define it.
Wait, so your criticism here is to say, well, they might have shown that for certain sub-regions of cortex, but they have not shown it as generalized.
They haven't found something that gives a unifying principle the way that one might have hoped. Yeah. Okay. Yeah, that's all I'm saying. I'm not doubting that there's been a lot of work. I think most of the work has been done towards the Hubel and Wiesel model, developing that. Not all of the work, but I think that that's the lion's share of the work. And I don't see any evidence, for example, that that's likely to explain language.
Oh wait, but we talk about anatomy here, right?
Yeah.
It's not about language. So that also means, but then the same.
For me, you could think of like people digging on two sides of a tunnel, and I'm worried that the people digging on the two sides of the tunnel are not going to meet in the middle.
Sure.
So I think that the people doing all the Hubel and Wiesel stuff are not working in a direction that is going to lead to something that's going to be adequate for explaining language. And I'm going to give a reason for that in a moment.
Sure.
The canonical circuit view also, I think, offers no real account of why there's so much cortical diversity. And there's a lot of cortical diversity at every level. Like John showed the famous Fellman and Van Essen diagram, and also the less famous Fellman and Van Essen diagram in his talk. And then there was work. Henry is an author on the stuff on the right. Is he an author on the Markov paper? I can't remember. And so on the right, showing things are a lot more complicated, and that's kind of the trend. And then there was a paper in Nature showing things were even more complicated, I guess, in the mouse brain.
Yeah, but look, now you're jumping out of the microcircuit, right?
I know. I'm going to come down though. Give me a couple slides and then ask a question. Then there is enormous complexity, obviously, in how the 6 layers connect to each other. This isn't displaying very well, but you should all look at this website, cognitiveconsilient.com, or better yet, download the app for this. It's a kind of amazing interactive diagram of what we do know about the 6-layer cortex. Then there's diversity at the level of neuronal subtypes. Of course, we don't even know how many neuron types there are, but there are hundreds by most people's count or more. There's complexity at the level of individual synapses. There's something like 410 different proteins identified in synaptic vesicles. And the point is, why do we have so much diversity if we're just doing one basic hierarchical feature detection computation? I like this. People like to quote from Ramon y Cajal with good reason. I like this quote from him. Unfortunately, nature seems unaware of our intellectual need for convenience and unity, and very often takes delight in complication and diversity. So I think that the extent of neural diversity is problematic for people that are looking for a few basic principles that, to explain the brain. It's not logically impossible that we could still derive a few basic principles, but to me, it makes me suspicious. It makes me think that maybe we want a slightly more complicated explanation. It also highlights the fact that there are many, let me just say, finish the slide and then I'll take a question. It also highlights the fact that there are many dimensions on which there could be important differences between cortical circuits. So one view is there's one kind of circuit. Another view is maybe there's a lot of different kinds of circuits, and the reason we have some of the diversity is to support that. Now take your question.
So it just seems unfair to jump these different levels and talk about all the diversity at the level of a synaptic vesicle, and then
the point is there's diversity all the way down. So whatever the level is that matters, we're going to find diversity.
That's right, but you're saying it militates against the view that seek a few simple principles. But you can have a simple circuit that's outputting the same thing with thousands of different molecules that could lead to that same output. In fact, the best work that's been done is by Eve Marder, who studies a very simple circuit, the stomatogastric ganglion. She can demonstrate that circuit can output the exact same thing with a 1,000-fold range of different ion channels, different neurotransmitters, so on and so forth. So I don't think just saying that, there's a diversity of proteins is enough to militate against the view that there are simple principles of the circuit level output.
Maybe I should have phrased it differently, but let's put it this way. The degree of diversity across levels of analysis is something that to me seems unpredicted by a view in which there's a single canonical circuit. Not saying it's absolute, and militate's probably too strong a word, I think I'll concede that, and I like your example from Eve Marder. But I still think it's surprising. I still think it's not what you...
I mean, I think redundancy in complex systems is a common thing. That happens in development. There's many different molecules. If there's an imbalance in one molecule, then other sets of molecules can make up for it so you can get the same outcome.
Okay, fair, but what's happening in development is that you're building a lot of different things. You're not building just one thing. So the other reason this doesn't really, well, I guess I have it on another slide, but it's okay. What I see evolution is doing is creating diversity over time. So you have duplication and divergence. You have families of proteins that do similar kinds of things but not identical things. And what I see the product of evolution over time being is family resemblance structure, not a single unified thing. So your vertebrae are different even though they're variations on a theme. your hands and feet are different, even though they're variations on a theme. And that's where I think we ought to be going, is trying to say, what are the families of circuit types rather than what's the circuit type? I don't know why. And then there's another hand. Is it Murray maybe in the back?
Is suddenly so worked up on this. But if you recall my introduction yesterday, it was very much the same. But I dealt with this in about 4 or 5 sentences. That that although you have if you squeeze your eyes, you can see some layers in all cortical areas.
Yeah.
But to really understand cognitive functions, you really have to look at the variability. And I showed some examples that you have huge differences in layer 4, 5, adapting to those particular functions. And all what Gary is saying here is if we want to understand these computational functions, we have to embrace these differences rather than looking for the similar. No?
That's mostly right. Yeah. I mean, I have a little more to say, but I agree with the encapsulation, and I nodded my head yesterday when you said that.
Just keep discussing anyway.
So I wonder whether it wouldn't be right to say that diversity is one of the simple principles. And maybe the other simple principle is to have some mechanism of selection among the diversity. I mean, evolution itself works like that. And personally, I suspect that that's very much the way cortex works, is because you have a rich variety of dynamics plus a learning mechanism which can select from among that diversity. And to my mind, I think that perhaps is one of the deep, simple principles that we might need.
Certainly Edelman would have it that way. I think that that's sort of an orthogonal line of discussion, and it's an interesting one. So, I mean, the one view is there's one circuit, basically the data sculpt that circuit. So you have some unsupervised learning scheme, and that drives you most of the way there. Another would be you have many different circuit types. I think there's a couple different dimensions here. Another is you have many different kinds of circuit types, and yeah, you do a kind of selective process.
Can I? Just a question. I think most people who are working on cortex work on primary fields like somatosensory, visual, auditory, because they're easy. Neurons respond really well and anesthetize and awake. And could it be, my feeling has always been there's something very similar about primary fields, and there's something very different about non-primary fields. So that maybe they are acting in a similar way with similar circuits. And then when you get into posterior parietal cortex or frontal cortex, you're actually making, my feeling is always that postnatal experience plays a greater role in those. So that there may be a diversity of circuits, but there could be groups of circuits that are quite similar. And the reason that we're coming up with this canonical view is because we're working in primary fields that may be doing similar sorts of things with sensory input.
I agree with like 98% of what you said. So another way of saying it, and is close to what you said at the end, is there may be a bias in the field because the primary sensory stuff is so accessible. The primary sensory stuff fits very well with unsupervised learning. It fits very well with pupil and visual. It's the most accessible stuff to work with, but there's been kind of a fixation as if that's representative of the whole. If there's one, I don't really disagree with anything that you said. There's a place where I might put some different emphasis. So I'm not sure it's just a function of input. I think there's probably some important molecular wiring differences between the prefrontal areas, let's say, and occipital areas, the sensory areas that we don't yet know about, and that we should be trying really hard to try to isolate. I actually agree that those areas are more plastic and depend more on input.
And one final thing, and I think, Zoltan, this is true, when even with studies of cortical development, people are concentrating on primary areas because we can identify them quite easily over and over again, or the barrels, things that we could see and manipulate. So, okay, that's...
Yeah, no, I totally agree. I think that's part of the underlying kind of facts about science and what can be done that drive the field in this way.
I have to add one word in agreement with you and disagreement.
He's going to carve a fine line.
And
fortunately, it's on the right side.
Just anybody who looked at Area 17 in a mouse and a tarsier would say they're different. They're strikingly different. So it's not that the same thing is done over and over again, even in a primary area.
I mean, the earlier you go, probably the more similar they are.
But Gary, at this point, I feel that your argument is bringing us to a point you're saying, like, well,
we have all this complexity, and I cannot imagine that this builds up to a canonical microcircuit or something
Well,
like
let me lay out my
But-
view.
but-
I haven't got
but
there
I
yet.
would like to push you a little bit more on that, because that's just not enough. And we should not get confused by variability. If you look in the room, everyone in this room looks different. But that doesn't imply that we cannot build a psychology or we cannot build an anthropology, right? We do share features as well. We have very similar, etc. Right?
If the diversity were the only argument that I had, I would take your point. But I think there are other arguments. So we can come back to that too. You can keep a running list of the things I promised to come back to and failed to. And then we have an hour on the podcast that you can really nail me down. Okay.
That's really brutal.
No, it's going to be in place of my lunch. It's terrible. Okay, so a third concern I have is that the canonical view, in my opinion, makes almost no contact with developmental, molecular, or evolutionary biology. I think it comes from neuroscience, and I think it comes from computational neuroscience, but I don't think that it actually makes contact with, for example, the tendency I already mentioned of evolution to for divergence and duplication, for building families of things over time. I just don't think that it touches that part of what's going on. Maybe a little bit of, probably it's not fair to say there's no reference to developmental biology, but there's relatively little contact between what I would take to be the canonical approach to neuroscience and the facts on the ground about evolution and molecular biology and how things are shaped over time, where things are modified over time, descent with modification. Okay, here's where, this is part of my answer to Paul's last question. If it were just diversity, things would be more up in the air. But then I think there are some computations that are outside the scope of the Hubel and Wiesel stuff. And this is really, for me, the core argument. I maybe trimmed too many slides here. Maybe I'll talk you through if you want. But since my first book, The Algebraic Mind, and even a little bit before, that book was in 2001, I've been concerned with a process that I would call variable binding. So in algebra, and I have this quote from William James, thought is a kind of algebra. I think James overstated the case. I don't think all thought is algebra. But if you think of algebra as a process where you have variables and you have instances of those variables and operations you can do over those variables, then I think that's part of what thought is. So in an algebra class, you learn that y equals x plus 2. X is a variable, plus is an operator, 2 is a constant, and you can stick a number into that value of x and calculate the corresponding value of y. That's what algebra is. I think that language can be thought of that way. I have an example in a moment. You have a sentence that's made up of a noun phrase and a verb phrase. You can put things into those variables and you do operations with them. If anybody's done any computer programming whatsoever, that's the essence of computer programming, is you define things in terms of particular variables and operations that you perform over them. Basically, almost every instruction in a programming language is something that you do with variables. And what I've been trying to argue for a number of years is that that's part of what people do. I don't want to say it's all of what people do. So I think you probably don't need variable binding in the lowest levels of sensory perception at all. But I think that you do need it for language. You do need it for abstract cognition. You certainly need it in order to understand a lecture like I'm delivering right now. And I think that that kind of stuff is outside of the domain of the models that have been presented in the kind of Hubel and Wiesel umbrella. This is the core reason why I think we need another kind of computation other than just hierarchies of feature detectors. And I've gone about trying to show this empirically in different kinds of ways. One is I took models that were popular in the late 90s, basically, but are suspiciously like some models that are popular now, and train them on certain kinds of regularities that are trivial for people and show that the models just couldn't capture them. So you train, this is the sort of Gertrude Stein example, you train people on sentences like a rose is a rose, a tulip is a tulip, a lilac is a lilac, and you say, how do I finish this sentence? A lily is a blank. And the model that was popular at the time, Jeff Ellman's simple recurrent network, was a model where you train it on sentences and it predicts the next word. So I framed this as a problem that ostensibly this very popular model is the most popular model in psycholinguistics could do, and it failed. So it would say a lily is, and had no idea what the continuation is. So there's a regularity that's apparent to people that this kind of architecture could not resolve. Another example was the identity function that I have in the middle. So your input is a string of bits like 0, 1, 1, 0, and that's your output, and you get a few examples like that. You can think of the ones on the top as being even numbers in binary. The one on the bottom is an odd number. If you take a multi-layer perceptron, which is at some level still the dominant model in the neural network community and a lot of machine learning these days, you give it any number of odd numbers, and now you, even numbers, and now you test it on an odd number, and the model fails to extrapolate in the way that people would. Tony has a question.
In the first example where you say that SRN will fail,
Yeah.
but it will predict a noun in that.
Well, what it will do, so the way that the output representations have to work in these systems, unless you want to break the model, is their localist output.
Yeah.
And what it will do is it will predict, it'll probably give some activity to rose, to tulip, and lilac, but it won't give any activity to lily. So does that count as a noun? Well, there's no tagged label for a noun. You can infer as external investigator and try to bend over charitably and say that it's predicted a noun, but it's not predicting the
It's
right
predicting
noun.
the
What it's doing is
yeah.
it's predicting many of the other nouns. But it is not, the crucial thing in this case, so I would also count a lilac is a tulip as a wrong answer. There's a co-reference relationship here. So you could put subscripts. You could say lilac X is a lilac X, right? And it's not getting that co-reference relationship.
What did you train it that, about a lily? Does it?
I don't think that one was trained in that particular case, but there's a whole family of cases like this. So let's go to the other one because I'm more confident off the top of my head. The middle case, which is formally the same structure, it's just not wrapped over time. So there you can do things like you can train identity for all numbers, and now you do string reversal for the even numbers, and it will still fail to extrapolate that to the odd numbers. I could come up with cases if you give me a couple minutes for the ElmanNet, but it's the same basic principle. The principle at work here, I think I dropped a slide, but maybe I'll, if I have it quickly.
But we could argue that the models were just bad.
Well, we could, but they're bad for a principled reason, and it's important, I think, to understand what that is.
Okay.
Let me unhide this slide.
We could also, think about what it involves for a person to solve this, because
We can, and let's go there, but second.
Technical challenges.
No, because I want to go through this one first. So the reason that these models can't do these computations is because each output is an independent classifier. So each output node is actually trained logically independently from the other. So when a network like this appears to learn an identity function, it's because it has learned individually for each possible output node what to do. There's no transfer between them. That's the basic problem with a hierarchy of feature detectors, is what you do is you tune up each feature detector independently of the others. I'm slightly oversimplifying, but not a lot. And so you get this phenomenon that I call output independence. And there's maybe one other slide that I'll show while we are there, and then we'll come back to how people do it, which is this one. The way that I think about it is in these kinds of models, you have what I would call a training space, which is basically a cloud of points around the set of examples on which you've been trained. I don't know if I can get this to animate. It would be better, but I can't. Oh, we have a pointer. So let's say I train you. Does the pointer work? Does it need a power? Power button. I got it. I got it, but it produced no response. 3 hours later, the power finally comes on, but only after the batteries are replaced. Oh, there we go. All right. So let's say I train you on 110 as your input. Well, there's some points that are nearby in space, and you can handle that. And then let's say I train you on others. So the green ones are the ones you're trained on, and then this is a kind of cloud of points around it. Well, you can do things inside the cloud. You can interpolate in these standard models between known examples. But then there are going to be some things outside that space, like maybe a longer number or a number where there's an odd bit that you haven't seen before. You can't extrapolate to those in these kinds of networks. That's the nature of the algorithm that is there. And I'm not saying there's no way to build any kind of model that does better than that, but if you think that what you're doing is learning feature detectors, well, each of these numbers, or each of those positions really, is a separate feature detector. And because the algorithm is local by design, There is no extraction of the general regularity. There's actually some, if you have hidden units, there's some interesting things that happen in the lower layers. But in the connection to the output layer, they're literally trained independently. So you wind up with this problem that you can't go outside the training space. So now let me go back a slide. So what do I think people do for these cases? What I think people do is they don't learn them as feature detectors. I don't think that your knowledge of language consists exclusively of relations between particular features. I think that's actually part of it, what you know. So I think we do keep track of the kind of stuff that a multi-layer perceptron would do, but we have another pathway. And that pathway is one in which we have representations of variables in which we can store, we instantiate variables with particular things. So you can say a sentence goes to a noun phrase and a verb phrase, which is kind of from an old version of Chomsky's linguistics, but it's a familiar, comfortable one. And so you instantiate it with something. So you have a noun phrase is the virgin mouse, which maybe is not a noun phrase that too many of us heard until yesterday. And then you have a verb phrase, like confounded Danielle's expectations, which even yesterday we didn't hear. She might have said confounded my expectations. We didn't hear. And then you produce a new sentence, like the virgin mouse confounded Danielle's expectations. And even though that sentence never existed in the billions or trillions of sentences that you've heard before, you have no trouble understanding it, understanding why it's funny in the context of this conference, et cetera, et cetera. My thesis is that no theory of cognitive neuroscience is going to be complete unless we can explain how we instantiate variables and perform operations like concatenations over those variables. So we can call it the Danielle test. Until we have a theory of neuroscience that passes the Danielle test, we are barking up the wrong tree. All right.
Barking up trees is good.
All right. And then the last, before I present my own view that's sort of been implicit all along, is that the canonical view offers no account beyond sheer brain size and maybe the nature of the input data for why language is unique to humans. I can't prove that that's inadequate, but my strong feeling is that that's very likely to be inadequate. That just saying brain size and functional input's not enough. So you could take the biggest chimp in the world, put it in the Nim-Chimpsky experiment. I don't expect it to learn language. And as I said, toddlers don't have the biggest brains, but they learn language better than even adults with bigger brains.
Isn't there a risk here that, don't you run the risk in your last criticism,
Before we go to your solution, that here you make a category error, because in some sense language is a complex functional property of a neural system, right? While the canonical view is actually very much dealing with the question phrased at a different level of description, which is what are the operations single circuits of physically defined, neurons can perform, right? So is this a fair criticism?
I mean, I think that this is the broadest one. I mean, this is I don't see the two sides of the tunnel as meeting kind of criticism. I think it's at the coarsest level of description, but it is still a concern to me. I mean, it's in fact the motivation for me. I guess I presented these kind of in the opposite of my own historical understanding of these arguments. I started with number 5, which is I looked around at neuroscience and I just could not figure out how to explain language acquisition at all with the tools of neuroscience. And then I thought about why, and that led me to thinking about variable binding and so forth. Anyway, so here's the conjecture that's inspired by a particular kind of digital circuit design. The conjecture is that we shouldn't be looking for a single repeated element that performs one kind of computation, but a heterogeneous set of what I'll call configurable computational blocks. Or I'll probably just call them blocks, where blocks are capable of instantiating a variety of qualitatively distinct computations, also some that are quantitatively distinct. So I don't mean to be imperialist about this, but to say that there are at least more, n is greater than 1, different, qualitatively different computations. There is at least the variable binding computation and the feature hierarchy kind of computation. There are probably more. I'll give you a candidate list later. But that there is a multiplicity. Each of those, in turn, might have subvarieties. And that any kind of complex action or language, I guess, isn't an action per se, or and it depends how you count it, is going to consist of the coordinated efforts of multiple blocks. So I should say here, I don't expect that we're going to understand language anytime soon. I would be really happy if we could understand some of the computational building blocks that go into language. I expect that language is going to be an incredibly complex circuit that consists of many blocks. But the place to make progress would be at least to figure out what some of those blocks are. I have a few more positive slides, but go ahead.
Yes, maybe you're going to get to this, but I'll ask it just in case. Can you define the scale of this computational block? Is it like within an area, or has nothing to do with an area? Is it a...
I am about to give a slide, but I'll just give an answer to that. I anticipate or understand them, let's say, to be either, but probably both. Some of them at the level of, say, multiple neurons interconnected. I'm thinking here of things like logic gates in computers, where a set of transistors comes together to do a particular computational function. I'm not thinking about them at the level of voxels in an fMRI scan. I think that that's way coarser. I think that one voxel would contain many blocks. But I'd like to hedge in the other direction, that sometimes what I'm calling computational block could actually be a single neuron. So I think single neurons may turn out to do very complex computations, and neurons may differ from one another.
But would a given area, like a cytoarchitecturally defined area, have all the same computational blocks?
My guess is that the areas, good question. My guess is that areas might differ in the composition of blocks that they have. So like a prefrontal block might have a lot more short-term, prefrontal area might have a lot more balance of say, memory blocks than a visual area or an early visual area might have, which might do a lot more feature detection. So some blocks might be rich in feature detection, some might be rich in sequencing, some might be rich in short-term memory and so forth. I think of them kind of like different Lego parts or something like that. So you can build a car, you can build a boat, you can build a house. I have a 20-month-old who's building these things. Hence the metaphor springs to mind. And some of these things are going to use more of some of these kinds of pieces than others. You're going to use the wheels a lot when you're building a car. But you can use the pieces in different places. It could turn out that some are exclusively made up of one or the other. Like maybe early visual area is just the machinery for feature detection and normalization and doesn't include any long-term memory.
I go with you with not having canonical microcircuits, but you seem to be buying in with at least one aspect of that, which is there's a considerable amount of modularity, and you can look at local areas in cortex and say there's some computation going on there within that local area. But you're talking about here a virtual machine which has these different functional components, but why imagine that these are particularly localized? They could be mapped onto the brain in many different ways, and a lot of the options would be very highly distributed, where the local computation in any particular area of the brain wouldn't make much sense.
I'm not committed to them being physically localized. I think it's easier to talk about it in those terms to get the idea out. But for example, some circuits might be involve loops to the basal ganglia or something like that. So they might in fact be pretty widely distributed. I think I have a slide later on where I list the possible substrates for these things, and you'll see some of them are distributed. That should probably be up there, and it's a good point. And the opposite of it is sometimes they could actually be superposed in the same spot. So you could have one circuit, and I think this comes closer to the Martin and Douglas kind of view, one circuit that actually does multiple things depending on what state it's in or things like that. So I think that full range is on the table.
But Gary, are you making a similar argument here like Rodney Douglas, who has actually used the notion of a canonical microcircuit and all their work on the anatomy of it to make the point that cortical circuits implement finite state machines that give you, if you want, an infinite space of computations to perform. So it seems you make a similar argument coming from exactly the opposite route.
Well, I do think that you're going to be able to assemble these blocks to do any computation. I don't really see the kind of quintessential block as being a finite state automaton, but it's not impossible. There might be some place at which these views converge. I think it's an interesting question.
Right. Okay.
Let me play it out a little bit more, but I think it's a fair question. So the inspiration, I said there was a digital circuit design that I like to think about, and that's the FPGA. I can just have a quick show of hands. I'm guessing maybe a tenth of the people in the room have heard of FPGAs. Oh, it's actually a significant number. So an FPGA is a kind of digital circuit that's basically a kind of computer that's laid out in parallel. Rather than having a serial step-by-step central processor, you have many individual logic cells, and each logic cell can be configured in a different way. And there's actually a family of circuits called programmable logic arrays, and I don't really care about the specific differences between them. But the idea is that you have basic building blocks that you can use in different configurations for different kinds of circuits, and that there are a lot of fundamental things that you can do. One way that people used to build these things is there was basically a bunch of different connections, and you could actually burn them out, like physically burn them out just before you would install one of these chips in a particular place. Now there are fancier ways to do it. Doesn't really matter for our purposes. But the idea is a given cell could do things like addition or multiplication or comparison, or could look things up in a table. So you have a stock of basic computations that you can do. You put them and you place them around this circuit, and then you have a parallel device that can do more or less any kind of computation that you want by configuring them in the right way. Question?
Precisely the other argument. Because all the logic blocks in an FPGA are exactly the same. All I'm saying is that all the logic blocks in an FPGA, are exactly the same.
Well, they are pre-configuration, but they're configured in different ways.
Oh yeah, you can do the connectivity in different ways, but the logic blocks are the same.
So
they're built of transistors.
Well,
the hardware, yeah.
I think that's it's sort of true, but misleading what you just said. So before you configure them, each block is a template that can be wired in a particular way, or I mean in many different ways. So let's just talk about the old version and not the most modern version. The old version of it, you have a bunch of wires, and you literally explode those wires. You burn out those wires. Once you have done that explosion of the wires, they are no longer the same. They are now each configured differently. One of them works as an AND gate, the other works as an OR gate. Those are logically different things. It's not fair to say that they're the same thing. It's fair to say that they are pulled from the same block, but it's like saying that I can do Michelangelo's David, right? I mean, I can buy the same rock, and I just have to dig it out of there.
No,
right?
Gary, but I don't think that's a fair response, because if you put your FPGA under an electron microscope, you will still see those wires. So in terms of the physical structure, the wires
under
are
EM, I think you could actually see the difference between the ones that are burned out and not. I'm not an expert, but
if you look at this, if you would take an anatomist perspective on that chip, you will see those wires, they will have a uniformity, and if you go deeper, you'll see that all these capacitors have somewhat different structures. These atoms are placed all somewhat differently, and there you have your variability, right? But still it supports a uniform computational infrastructure.
Well, yes and no. So I don't think that's entirely fair either, even though I take a lot of what you said there. So in fact, I like this observation from the opposite direction. So the way I would put it is that, yeah, if you looked under a magnifying glass, an FPGA looks perfectly uniform, but that would be a mistake. That wouldn't capture the essence of how it's doing its computation.
Sure. But however, you criticize the microcircuit, which is purely defined in structural terms, also on those grounds. You say the microcircuit as a structural, physical expression might not hold, right? I mean, now you criticize it on functional grounds. So we should keep these two levels of description clearly apart.
Okay, but if you go back to the FPGA, once it is burned in, the configured as opposed to the pre-configured FPGA, once it is burned in, there are anatomical differences, literally, that correspond to its functional differences.
And this maps onto development, so you can say that the
Exactly.
original FPGA maps onto the equipotential cortex, which isn't fully wired up yet. Yeah.
Exactly. Let me put up this next slide, and then I'll take more questions. Because I wanted to get the molecular in there somewhere, the molecular cues and developmental side. So this is kind of a caricature of a few possible positions. One is you've got canonical cortical microcircuits tuned by experience, and that's part of what I want to emphasize here. The anatomy is essentially uniform. The computations are identical, only to the extent that they're tuned by different inputs, and the wiring is tuned by experience. Then there's an fMRI literature on functional specialization that's really actually pretty vague about what the mechanism is. People just say, this area does this thing, and they don't really say how or why. But it's often implicitly presumed that the underlying anatomy is heterogeneous. The computations are presumed to be heterogeneous. That's supposed to be what that literature is about. And nobody specifies anything about the wiring. And then there's the position that I'm trying to push, which is that the cortex is an array of reconfigurable computational elements. The anatomy is largely shared, just like the anatomy of the FPGA is largely shared across its extent, but there's some molecularly guided fine-tuning for the individual blocks. The computations are tinkered variations on themes. Not saying that there's an infinite array of these things, but there's an array of them. They're variations on themes, and they are pre-wired by molecular cues and shaped by experience. And that, it's the first half of that maybe that I think is most left out of the kind of computational neuroscience. I don't see the molecular cues in the computational neuroscience except in the special case of people doing the developmental biology of topographic maps. So people that specifically study the computational neuroscience of how you get topographic maps worry a lot about molecular cues. But I think people that build Hubel and Wiesel models don't worry about molecular cues at all. And they don't worry about, if you had molecular cues, could you build such that you had different logic blocks that did different kinds of things? Now I will take questions. This is a good place to take a question. First one today. No, but anyway.
In fact, it's so good place
So
that
now we're a quarter of the way through.
I completely agree with you with your last column, and I would go even further. Yesterday, perhaps we did not really emphasize some of the transient circuits. When the cortex is constructed, you have some of the afferent projections already developing on a platform which is called the subplate. And these are largely transient, early generated cells, and they help to program some of these area and region-specific circuits, and then they get cleared away. So they are largely transient. So actually, I would almost completely agree with your last line.
Yeah, I mean, another way to put it would be there's a lot of work in developmental biology, in the actual processes, to do programming. And it's not reflected in the computational models that people are building by and large. There's clearly, if you look at what role the genes are playing, it's to help program intermediate stages, build scaffolding. We don't have a neural tube at the end of the day. We have something that, but we have lots of steps along the way that are there in order to sculpt something in a particular shape. And not every step there in that development is reflected directly in the end. Some of it is scaffolding to get you there. But you ultimately want a developmental neuroscience component for your computational model. You want to understand what is the role of all of this sculpting.
And if we look at these transient scaffolds, it's also interesting that you have a very heterogeneous cell population. And if you start to look at area differences, even this pre-programming of the cortex, so this transient circuit is different according to the different areas.
That's right. There are gradients, for example, that are clearly important. And I'll come back to the gene expression differences a little bit in a coarse way.
So I think one of the things that you mentioned it in your previous answer is that it's not just cortex. So cortex could be the memory component of this, and then nearly all of cortex, except some of the primary sensory areas, is in a loop with the basal ganglia, and that could be giving you some of the sequencing, some of these other properties that you want from
I totally agree with that.
But that would allow for cortex to be doing something fairly uniform, but sort of, as you say, tuned to the different functions of the large circuits within.
Yeah, and I think that it could be, if you wanted to stick with my FPGA analogy, but also take on board your distributed notion, it could be that what you're doing there is partly tuning, saying, I'm going to go off to this other. So I'm going to tune this particular area to invoke the basal ganglia in this way, as opposed to this other area. And so I think there's room for some kind of unification of those two strands.
So the people that study the basal ganglia, I've done some work there myself, I would say, again, there's a canonical microcircuit in basal ganglia. It's doing a different kind of computation to cortex. You put the two together and the whole system then is a bit more like your FPGA.
Yeah, but why not take that a step further and say, look, we know that there's a different, not that anybody knows exactly what any of these things do, but we have a pretty good guess that the cortex is doing something different from the basal ganglia. But why think that the cortex is doing one thing rather than think, oh, well, maybe there are a bunch of things going on here that are some family of computations. Like, at some level, I don't understand even that instinct other than out of parsimony, and I don't think parsimony is such a hot idea in biology.
But then your summary here of your position is a perfect summary of the work we have published on these objective functions that generate visual hierarchies. Okay, it's all there.
Including the pre-wired by molecular
Oh yeah,
cues?
completely, because it's not that it starts just with a clump of cells that build themselves up in hierarchy. No, there are predefined. There's a predefined layering that is required. There's a predefined envelope imposed on receptive fields or the projective fields between these layers. There's also pre-specification in some sense in terms of time constants and so on. There is pre-structuring, absolutely. Otherwise, this doesn't work. And then that's shaped by experience. It's a perfect summary of that kind of work.
We should walk through some of that after, probably offline.
Are you going to talk about the pre-wiring by molecular cues? Because the problem you have is that you said at the beginning you've only got 5% of the DNA because the 95% is shared with chimps.
Yeah.
And then you've only got 24,000 genes, so you've only got a few thousand genes to wire up your cortex with.
Whoa. Again, I stopped.
Well, okay,
a number of your questions have this form where you start with three things I agree.
But yeah,
you have all the genes to shape your cortex with, or nearly all of the genes.
But you don't have...
You don't have that many to share the humanly unique part.
Well, exactly. So you've got to take existing gene networks and use these for configuring your array here.
But then you can tweak those. So, I mean, the way I think about it is that you've got a whole library of subroutines that you've inherited from our last common shared ancestor with chimpanzees. There's a whole library of developmental subroutines for wiring things up. And what we're doing is we're taking those subroutines and doing something a little bit different. But it's the whole accounting is a little bit weird because if you're at the top of a subroutine, you can do a hell of a lot by changing one line of code or something like that.
Yeah, I think I agree with pretty much everything you're saying, but the shape by experience has got to be important here because, like you say, language appears in, it could be 50 million years, it could be,
Well, the empirical range is 50,000 to 700,000.
Yeah, so 50,000 to 70,000. So that's not enough time for anything genetic to really change dramatically.
I disagree with that for reasons related to what I just said a moment ago. So, you think about sickle cell anemia. You can have a single nucleotide change that has a dramatic consequence. I think that language is a small number of genes with a dramatic consequence. I think the genes are important because those are genes that are at the top of cascades. So FOXP2, like PAX6, is at the top of a cascade, and that's part of why it has as much consequence as it does. So if you have mutations in the right place and they're speaking to, they're sort of like CEOs, that they're shifting the direction of the entire company with, their one sneeze has a large consequence. All right, let me go on. There's only so much time, even with our liberal presentation format. I'm not that far from the end, believe it or not. So maybe that was a lie. I think there could be many different kinds of blocks. All of this is just sort of a point in a direction. So I'll point a little bit more in that direction without committing. Some of your hypotheses for what those blocks might be ought to come from the kinds of bottom-up work that people do in experimental neuroscience, computational neuroscience. So I think there's good reason to think that there are topographic maps, lateral inhibition, and so forth. These are all going to contribute to the kinds of blocks that you have. I think there's lots of top-down stuff that sometimes gets left out of neuroscience. I think sometimes, I heard one neuroscientist say, data talks and theory walks, and I cringed on behalf of neuroscience when I heard somebody say that. I think there's lots of good reason from theory and cognitive psychology and so forth to think that there are things we can't yet identify in neuroscience that are actually really important, like short-term memory. So we don't yet know the neural substrate of the kind of short-term memory where I say, let's change our podcast thing to 3 o'clock instead of 2, and you show up at 3 o'clock, one trial. We don't know the substrate of that, but there's got to be a block that does that. And I think there are a bunch of blocks that I have on the right that I think would be critical for language. We don't yet know the neural instantiation of them. Working memory storage, long-term declarative memory storage, variable binding, copying and pasting things into working memory buffers, machinery for orchestrating sequences over time, coincidence detection, storage of temporal sequences, processes for flexibly gating information. The gating information, there's been a little bit of work recently by Bill Newsom and others, maybe other people I don't know. I see the candidates for blocks as coming from both lines of research, both from the kind of bottom-up research and the top-down research. My guess is that all of these things are instantiated with different kinds of neural circuits, and that we don't yet know how to identify these things. If we did, we could start to attack the higher level processes. So how does a parser work, or a sentence parser, Or how does an object scene decomposer work? Well, I don't think we can get there straight from neural circuits. I think what we need to do is to identify all of these kinds of intermediate blocks, and then we have a chance at understanding the larger things. I had a slide about configuration being a mixture of molecules and activity. I don't know that I really need to go through the slide, but I think that obviously experience is really, really important. Nobody should deny that. I'm not trying to deny it. But I think the molecular stuff is getting undersold in people's computational models of how the adult brain works. So I assume that to configure a block, you have available the entire toolkit of molecular biology, which includes things like cell division, cell migration, axon guidance, and so forth. Sometimes I think about this whole project in terms of synthetic biology. So if you were going to build an FPGA out of biologics, what would you use? You would start with the toolkit of developmental biology. You would also use activity-driven mechanisms. I'm going to say a little bit more about that in a second. And duplication and divergence. If you were doing this, as an engineer, you would use that a lot. You'd say, I can build a family of these circuits by tweaking these kinds of parameters or making a copy of this, combining these two things, and so forth. And I'm guessing that biology has done exactly that. If you look at any other domain of biology, that's what you see. So if you look at the developmental biology of the limb structure or the heart or the circulatory structure or so forth, you see duplication and divergence. You don't see from scratch development where it's as if an intelligent designer said, what's the best way to solve things? You see, what's the best tinkering that, you sort of get local maximum where something works pretty well given the set of pieces that are out there. Okay, some reasons why I think you should take my conjecture seriously, despite the fact that it's obviously ill-defined. One is, I think it gives you a shot at explaining functional diversification. I really don't think that the Hubel and Wiesel model is going to get us to the diversity of higher-level human cognition or language. I think it's really good for categorization. It does well with speech recognition in practice in the AI field. It does well with image recognition. But even within vision, it doesn't do very much with, say, analyzing a scene. It doesn't do anything for planning, doesn't do anything for language. So we have some idea of what the Hubel and Wiesel model can do as an engineering artifact now, because a lot of people are working on it. They're spending a lot of money, probably eclipsing the academic budget for these things by a factor of 10 or 100. And those are the kinds of things that they can do with them, is categorization. They don't know how to do these other things. So Google has probably spent literally a couple $1 billion on deep learning in the last couple of years, and they have not been able to get it to do their natural language understanding, for example. Functional diversification, where you could have different circuit types in different areas or different collections of circuit types in different areas, at least gives you a possibility, I think, of approaching that problem. And then I've already alluded to this compatibility with developmental and evolutionary biology. It gives you a way of thinking about, well, we know certain things are true, like that duplication and divergence is a critical property of evolution. The reason I came to this conference is in part because I love John and Leah's work on these kinds of things, duplication, divergence. We have already knowledge about how this works at a kind of coarse grain, but it's got to work at a fine grain too, and I think this is what we want to figure out. I think that we want to make use of the fact that there can be qualitative differences that emerge from evolution, even if they start as minor parametric tweaks. So the hand versus the foot are, in some sense, minor parametric tweaks on another. And at another level, they're really different things that have been tuned to do different kinds of functions. And I expect that we should find that in our computational building blocks. I see no reason a priori to doubt that. I don't think that the uniform architectures really refer to this hallmark property of evolution about duplication and divergence. Okay. Possible way, it also offers a possible way to think about human evolution. Obviously, much of what any individual knows is learned. We have education, we have culture, we have the internet, wherever that fits in, now that we're at the right end of Via's figure. But we also have a gift for acquiring deeper abstractions than other creatures do. So it's not just that we are exposed to cultural institutions or to other people talking. So you could raise chimps among us, give them unlimited internet access so that they can watch all of the internet videos on how to talk, and it won't get them there. I think that having new circuit types mix in mostly with ancestral circuit types might give us that edge. What we're really trying to explain here is why our learning mechanisms are different from chimpanzees' learning mechanism. Why is it that when we are exposed to language or law or philosophy, that we come away with something, and when chimps are exposed to it, they just want to know where dinner is? I mean, part of it's a motivational thing, but part of it is some kind of representational and algorithmic ability to manipulate those data when they come in. I think knowing about different circuit types might give us a little hint of that. So I have a few more slides. Are we doing on time? We're good. Okay. You'll give me the hook when I need to go. I guess there's a slight problem in how I put together my talk. I'll come to this one later. Let me give you a little bit of apparently conflicting evidence. One is this Hill et al. Study, which I mentioned before, that says basically you take S1 from a bunch of different rats, you look at where the synapses are in 9 rats, you can predict where they'll be in the 10th rat just by knowing where the neurons are, basically. And if that were true, their summary is random alignment of axonal and dendritic arbors provides a sufficient foundation for specific functional activity to emerge in local neural microcircuits. If that observation, which was in PNS, were true in general, then you wouldn't need the kind of theory that I'm trying to push people towards. So if it were true that there's simply a generic architectural way of wiring things up, then I would be wrong. But in reality, I don't think it's nearly as strong a test as this paper portrays it to be. So one problem is there's no test there actually of how this work that they did generalizes either to other cortical areas or to other species. So it could be true that you can predict S1 within a rat, there's some consistency there, but it doesn't even generalize to the next area over in a rat. And it's also possible it doesn't generalize at all to the next species over, and they just don't have any test of it.
Gary, in this paper, they make the point that this is like supporting these ideas of, for instance, Jerry Edelman, that a number of processes are operating on these initially defined circuits to give you certain functional properties. I don't think the argument would be like, and this is it.
Well, I mean, I think the modeling is sort of premised on it. I don't know what their real takeaway is. I think the modeling pursued is premised on the idea that you can predict the circuitry of the brain by taking relatively modest sample sizes, fully understanding the connectivity there, and then guessing it'll be the same. Now, if you look carefully at their data, I don't remember if I have this on a bullet point or not, but they actually only get about 75% of the variance. So it also raises the question of maybe what's really important is the last 25% of the variance. So one possible view that's somewhere in the middle is there's a canonical base frame, and then there's important variation, some of molecular, some of it based on input, and that's what gives you the whole enchilada. And they're telling you something about the canonical base frame there, but to really predict what's going on, I mean, to really understand what's going on, you need to understand the differences between different areas that build on top of that frame. And probably a lot of people would be sympathetic with that. I think that's a reasonable view, but it is one where you need to know the wiring cues as well as the input in order to be able to say that this is why this is doing this function rather than the other. Question, or can I?
It's just a comment, really. I think all this study tells us is that S1 barrels in one rat is a bit like S1 barrels in another rat. I mean,
and it's not even true. I have at the bottom. So it turns out that the cellular organization is actually whisker specific.
Yeah, exactly. That's right.
So it's not even true there.
So in the whiskers, it's probably true. But I mean, I think it's also probably true that the specific alignment of where the synapses are in any part of cortex isn't going to be specified. That's going to be some self-organizing process, but it's not going to be uniform, as you're saying.
It's not going to be uniform. It's not going to be entirely self-organizing either, on my view. On my view, it's going to be molecular plus input. And so
well, you think you
different
have to give us some
molecular
examples of how these molecular processes are going to do all this clever wiring for us.
I'll get to that in a
Good,
second.
okay.
They also give no account of the long-range connection. So even if your inclination towards distributed connections was 100% of the explanation, they would still have a problem, or even more so. I mean, if it turned out that what really makes one cortical circuit different from the next is how it connects in long range, then they have nothing to say about it. They're only actually looking at the short range connection. So I don't think it's actually a very compelling argument. Then there's the rewiring experiments. I kind of already talked about this. they actually only been done within the sensory areas. It's not really a general demonstration that there's cortical interchangeability. As Paul points out, evidence of absence and absence of evidence are not the same thing, but at least we don't have any evidence on the table that suggests true interchangeability of cortex. So if you took this as evidence for that, you would have a counter-argument, but it's not really. I'll skip that one for now. And I will also skip that one for now. So let me give some evidence that's consistent with our view. One we've already alluded to, might have been Tony who asked many hours ago when I started this talk. And John showed data like this. Or no, who was it? Asif, who said Brodmann areas, right? I mean, the fact that you have Brodmann areas is actually nice for this view. It actually kind of possibly fits. There is some evidence at the electrical level that different cortical areas actually operate differently. There's less evidence one way or the other in this direction, but here's something that just came out that kind of points in that direction. Then, this is something I said I would get to presently. If you care, I mean, on my theory, maybe I haven't quite fully spelled this out. What you're looking for is molecular codes that can help you to wire circuits differently in different areas. That doesn't have to be the final draft. You can think, what I need for my theory to be right is that you can make different rough drafts in different cortical areas of different circuits that are going to respond to their environment in different ways. If you can only have one rough draft everywhere that's uniform throughout, then you have a canonical cortical computation, and it's all experience. The alternative is I can wire up rough drafts in different areas, and they get calibrated differently depending on which rough draft I have in which place. This is how I read the developmental neuroscience literature. There's a very nice paper from the Allen Institute last year showing that different sets of molecular cues can actually take mouse S1 layer 5 projection neurons, which are kind of a canonical case that people look at, to send them to different places. So this is about the long-range connections, but it's a molecular cue system that could allow different cortical circuits basically to work differently by just targeting different external areas. So invoking the basal ganglia or the cerebellum or whatever as different kinds of subroutines. So that's one kind of tool, again, that at least if I were doing synthetic biology that I could lean on. I could actually take this existing code, if I wanted to build a synthetic FPGA computational device, I could take this existing code as a way of having axon guidance processes and so forth that would target the right kinds of areas and do it differently for different cells that were carrying different sets of genes. Yeah. The Sorensen paper.
But this excellent, very nice paper is actually not providing us with a toolkit how to wire up the brain. It is actually just a pure correlation between gene expression and target selectivity. So these are just the first steps to understand that algorithm which will generate that specific connectivity. But I think it is a good illustration that eventually we should be able to do this. Yeah.
I mean, it's possible to be sympathetic to me, even though I agree with what you said, that those are the targeting molecules in the best case. Or maybe it's not even, if you know the molecules well enough, maybe you can rule that out. I generally agree. It's correlational. It's not cosmetic. Then there are alternative splicing molecules that could really be interesting. So if you think that a lot of the work is done at the kind of last mile of how synapses are wired up, so you have a canonical frame that is then tuned in different ways to do different kinds of circuits. Much of the action might be at the synapse level. Alternative splicing is a way in which you could get individual synapses to be doing different things, basically. And there are some candidate molecules that actually allow you to unify the molecular cues with activity dependent, where they're responsive. The splicing itself is responsive to activity and to molecular cues. So you can sort of the logical conjunction of the two. And so that's at least a kind of mechanism that you would want. All right, last few slides. This time I really mean it. How should we move ahead? What should we do in neuroscience next? So this is really zooming out. Everyone agrees that we need new techniques. So I mean, there's all this discussion because of the European Brain Initiative, the American Brain Initiative. There are going to be some in Asia, I think, as well. A lot of money is about to be invested in neuroscience. How do we want to spend that money? What should we do? Well, the techniques are obviously critical. We want, I think, on anybody's account of how we proceed, we want better ways of measuring neural activity at the level of individual cells. I think we can all agree that looking at the level of voxels is not really getting us to understanding how the brain works. Maybe helping a little bit, but it's not the final answer. We need to be able to do things like record from many, many individual cells simultaneously. Everybody agrees we need to take the ideas of optogenetics and make them more practical and extendable and so forth. Given finite resources, there are a lot of different things we could prioritize, like the original proposal for a brain activity map, which led to the Obama initiative, but was actually kind of partly abandoned along the way, said what we want to do is to map the neural activity of the entire brain. Rafa Yusta is fond of this analogy. He says, think of the brain as like a television set. We're looking at one pixel. We need to see the whole thing. I don't actually happen to agree with that argument. I think if we could look at like 50 pixels really closely, maybe that would be great. We don't need the whole brain, but that's an argument for a different day. We might try to map the full connectome, which is, of course, a big job unless we find a way to automate it, like Tony Zadar is trying to work on. But for now, the only way we can map the full connectome is to do a lot of EM and a lot of reconstruction that requires a lot of human labor. Tom Dean, my collaborator, is actually trying to automate some of the EM reconstruction. We could try to simulate the entire human brain, as your Swiss colleagues allege that they're trying to do. You could try to exhaustively map a specific area, which is basically what the Allen Brain Institute is trying to do. They're trying to map visual cortex in a mouse exhaustively from retina to as far as they can get. You could catalog all of the individual kinds of neurons. That's one of the goals of the Obama Brain Initiative. What I would suggest is that what we want is a taxonomy. I'm going to give you an example of a taxonomy in a second, of mesoscale computational elements, where what we really want to focus on is bridging. We want to focus on bridging between computation such that a cognitive psychologist might describe them or a computer scientist might describe them, and neural circuits. We want to know what physical realizations are there of computations like the things I was suggesting the blocks are. And I think that points to prioritizing two things. One is comparisons between different cortical areas. So I think that just figuring out visual cortex is not going to get us to understand the array of blocks, because I think visual cortex is mostly going to be about hierarchical features. And as I've said, I think that's important, but I think it's only part of, well, and especially in the mouse. We're going to put bifocals on them and see if it helps. And then I think it points to co-registering activity, transcriptomes, proteomes, individual proteins, and so forth. So doing comparisons between cortical areas and looking at them from many, many different techniques. So not doing the whole brain, but doing some focused comparison, trying to understand what is different about a circuit in prefrontal cortex from a circuit in visual area. The goal is to make a table like this, and I know you can't read it if you're sitting in the back, and maybe not even if you're in the front, but I'll kind of point you through the gist of it. And our intention is actually to put this on the web, although we haven't yet. In the left column is a set of computations. And this is basically straight from David Marr, for those who know Marr's work. Then there's a column for algorithmic realization, for neural implementation, and for brain location. And then the rows are things like rapid perceptual classification. So these are the computations. Complex spatiotemporal pattern, something that got cut off, and I will reconstruct it later for you. Working memory, decision making, gating of information flow, gain control, sequencing of events over time, variable binding, and so forth. And so the idea is that we want a matrix that says these are the computational building blocks that we think that the mind might plausibly be made of. These are the ways in which they might be implemented computationally. What kind of computation could give you variable binding or could give you gain control? And often there are candidates for some of these things. And then what are the neural implementations of these things? I think that, and where are they? And going back to your point about distribution, some of these things are going to be either the computation itself is going to be distributed, or they're not all going to be in cortex. And in order for cortex to do these things as a whole, it's going to have to call these subroutines elsewhere. But I think that our goal should specifically be this. I don't think our goal should be we want a map. I think our goal should be we want a matrix that bridges these things. So closing words. If language is indeed an orchestration of something old and something new, as seems at least virtually certain to me, the best place to start might be by systematically trying to identify computational building blocks that we've inherited from our pre-human ancestors, and then eventually seeing how a small number of human unique computations and human unique formats of representation interact with the rest to yield a creature, not a capable of acquiring a vast array of complex systems of knowledge, from language to music to science, and I thank you very much.
Okay. We have time for just a few more questions.
That was about twice as long as my average
know.
talk, so I thank you all for your patience.
You're slowing down, Gary.
Hold on a second.
Hi. Just one question. So we know that monkeys, chimpanzees actually can speak sign language, but they have a problem with grammar. So for example, banana eat monkey and monkey eats banana. It's for them the same thing, right?
Yeah,
So
I mean, the literature there is complicated. There's one animal, Kanzi, and actually Kanzi's daughter, I believe, that allegedly can do more. But in general, I agree that that's the upshot of that literature. I'm suspicious of the Kanzi work. I can tell you later why. So in general, the animals can't do syntax. They can do association with individual signs, not even sure that that's the same thing as a child learning words. So I have a 20-month-old. I watch him learning words. He does this often on between 1 and 10 trials. And Kanzi needs many, many trials in order to learn things. So there's association to learn a word. I'm not sure that's the same thing as a human learning a word.
yeah, so the thing is that I understand that, for example, grammar is something that could be more unique to humans, let's say. And if we think that grammar is some sort of action sequence, what is your opinion in terms of, I don't know, premotor cortex? How could that contribute somehow to this evolution of language in terms that humans are so good at language?
I think there are a lot of different possibilities there. First thing I'll say is I don't think that grammar is just motor action sequence, but I think it could have borrowed from motor action sequence. So I think that there are a lot of things that go into grammar that don't go into motor domain. Like you have to get co-reference right. So if I have a pronoun later in my sentence, it has to link, or a reflexive pronoun later in my sentence has to link to the front of the sentence, let's say, in the proper way. Maybe you don't need the same thing in the motor domain. But you may need some of what you need for language in the motor domain. I think motor domain is a really good place to look for candidate mechanisms that might have been borrowed, because you do need sequencing. You may even need hierarchical sequencing, which is obviously critical for language. So I think some of what's in the motor system is borrowed, but then there are other components that are not. And then language, for example, uses a lot of theory of mind, your ability to anticipate other people's needs and so forth, that don't per se have anything to do with motor. So I think there's a borrowing of that toolkit, let's say, and maybe an extension of that toolkit, and that's part of what language is.
Can I,
we have a guest from outside, at least from across the street, Luca Benatti.
Oh, this is just really brief in your kind of matrix. You didn't really speak, and for me, I think a large problem of trying to understand these complex things like language is you didn't really talk about bridging different levels of hierarchical organization, from genes to molecules to brains to behavior. And I mean,
all I did is to talk about filling in between brains and behavior by having a computational set of building blocks in between.
Because people are going from molecules to behavior or genes to behavior, really
I think
complex
people
behaviors.
are reaching too far, and without this intermediate, we won't be able to make those bridges.
Right, but in your talk, I didn't get any of that sense that we should be doing these sorts of things to understand how humans are different than chimpanzees. And I think,
I'm sorry, I don't think I understood the question.
well, you talk a lot about computation, but not about how are there ways or is there some sort of computation where we can understand these different levels of organization? They're operating on a similar principle. I'm probably not phrasing my question very well.
Let me sort of explain how I see it, and then you can come back at me. The way I see it is we have an underlying physical architecture, which is basically neurons configured in particular ways. Then we have behavior, and something intervenes in between them, which is computation. So we don't go straight from brains to behavior, or maybe a different way to put it is that behaviors, I think, are dependent on intermediate level building blocks. So you couldn't program Microsoft Word if you didn't have AND gates and microprocessors in between having transistors. Like there have to be different layers of complexity on which things are built. And I think that behavior is built on top of having intermediate building blocks that do computations. Those intermediate building blocks are physically realized, and the physical realization comes from the conjoint action of genes, experience, etc. What I'm saying is we need to understand how these intermediate blocks are built in order to understand how the more complex things are done. And really, there's even more levels than I'm saying. So any particular behavior is going to depend on the action of multiple brain systems. There are very few behaviors, except reflexes, that don't invoke many systems, especially in humans. So you have systems that are deciding your course of action and remembering things and so forth. They're all interacting in behavior. But even those individual systems, I don't believe that we're going to be able to understand them directly from connectomes unless we recognize, another way of putting it that I didn't use, is motifs that represent particular computations. So they're going to be intermediate levels of architecture out of which the more complicated things are built. I think that this is true elsewhere in biology, it's true elsewhere in computation. So much of computation is about building different layers of interfaces and so forth. So you have transistors, then you have logic gates, then you have microprocessors, then eventually you get to things like internet stacks and so forth. Each of these things is built in a lawful way upon the others that are below it. And it would be absurd to understand how my laptop works at the transistor level without understanding some of those intermediates. It would be absurd to try to understand a laptop without understanding a programming language, and you wouldn't really know what a programming language is, or wouldn't fully understand what a programming language is unless you had at least some basic concept of the logic gates that are within. And I'm saying we're not going to understand behavior unless we understand the kind of components that go into behavior, but the larger components like parsers and decision makers and so forth. But even those, I think, are way outside of our grasp. They're too complicated for us to be able to directly map them. But there's some intermediate levels of organization that are doing computations, and that's what we need to go after. Does that help? You can come back, and Luca. They've had their turn. This is your one question. They've been asking questions.
No, well, the point, I didn't pay to be here. I don't know what I'm allowed to do. So I didn't need the
You're a guest of the
so
speaker.
Right now you're allowed to pose a question.
Okay, so lots. I mean, I'm not sure I follow fully your talk for lack of knowledge about the importance of each of the questions that you raised. But lots of your background comes from specific feature of language, and as you know, I very much agree with the fact that this is really an important thing to be looked at.
You've made comparable arguments, I should say, with respect to reasoning, for example.
That's what I really wanted to ask you. If you look at language, it's really, I mean, for what people know, I know very little. I certainly don't know anything about the brain organization of language. I don't know anything about the brain. But if you look at the role of experience in language, after all, it looks like Chomsky has been vindicated that the role of experience is really peripheral in a very important sense. So if you look at, for example, 3-month-old premature kids, their circuitry, even before any migration of the neurons to cortical areas has been developed, their circuitry seems to work like adults. If you look at how much experience you need to do things like, for example, phonological narrowing, which is a classic case of experience learning, it looks like experience is not very much important, because if you look at premature babies who have like 3 months of experience more, they don't change the way the moment in which they converge towards their sounds of their natural language. So it looks like there is a lot of structure, let's put it that way, and the experience really does the tweaking, but what we really need to understand is the structure there. Now the question I have is...
Can I just pause to kind of bring people who aren't from psychology up to that with something that they would follow, which is a lot to people in developmental psychology like Luca and myself, a lot of the language literature is reminiscent of imprinting, where there seems to be something that very specific that is targeted and can be acquired very quickly. So there's some learning in imprinting. You see, Conrad Lorenz, and you learn that that's your mother, or at least the closest substitute to your mother, and you imprint on it. But there's clearly a mechanism in place to allow you to do it. It's not like you need 1,000 trials to do it. You're looking for something specific, and it's targeted. Seems like the same thing is going on in phonology. You don't need infinite amounts of experience to learn the specific sounds of your language. It seems to be a little bit like imprinting, where it's very directed, where you don't need a lot of experience to get you there. And it's like you're choosing alternatives that are
And where the molecular mechanisms which are involved are really important, because we know that critical period and imprinting depend on really, they know, I mean not me, but I can only read, really depends on exactly when the experience is being delivered and what is your, what molecular consequences receiving the experience has in that particular moment. So the question I have is,
that part I think is actually weak.
well,
I mean, we could talk about that separately, but I don't think the critical period literature is as strong as people thought it is, and I don't think it's as developmentally or biologically clocked as you might think it is. I think that there are factors like interference that are psychological factors that do vary.
well, unless you think that these factors that seem to modify the critical period are really factors that modify the underlying molecular reaction.
We'll adjourn that argument.
Okay. So the question really I have is, there is this literature that says, okay, what are humans? Well, humans are chimps plus language, right? And in a sense, that's what people like Spelke are saying, right? Really what counts to explain the difference is language. And now, then there is you who say, look, language is, for lack of a better word, a kludge, sorry, of putting together things that are really not necessarily for language. So, but we do know also that humans have a lot of other things that come together, like reasoning and so on. We also know that if you have a good language, you also have good math. I mean, so how much do you think that your theory is dependent precisely on the fact that we speak language, and how much, what range of organisms could be possible given the kind of early circuits that you think is available?
Having watched my own older son, who's 20 months old, I'm a little bit of less nativist about the specifics of language. I guess for the last 15 years, I've become less and less convinced of domain-specific circuitry, but still convinced of innateness. So I think that there is innate circuitry for sure in my son that makes him good at abstraction, for example, much better than current learning algorithms are. I don't know how much of it is specifically tailored to language. You could look at least at the examples of people that learn, say, sign language as opposed to spoken language. You see that there's some modality independence. So there's a lot of things that we do that might or might not depend on language. So we can learn abstract systems, like maybe you could learn to play the game of checkers if you were human, even if you didn't speak. I'm not sure of it, but I think it's at least within the realm of possibility. And I don't think it's possible that a chimp could learn to play checkers, although someone's going to go out and do the study and refute me. But I think that what's really special about our minds is the ways in which we can compile new abstractions. And not just abstractions like the feature hierarchy kinds of things, where you say that a letter is more abstract than a line or something like that. But like we have whole new sets of representations that we can manipulate and use abstractly and build new things out of. So we can construct the notion, at least of a written word. That's one that isn't innate. The notion of a written word is something, an orthographically spelled out word is only 5,000 years old. It's not universal, but it clearly taps into this mechanism that allows you to build new representational architectures and assign them meaning and so forth. It's possible that whatever machinery allows us to do that also allows us to bootstrap language. I'm not saying that that's certain, but I think that that's maybe on the table.
So it's just an accident of how the literature went, that all the features that are really important to build your case from a psychological point of view essentially come from language. It couldn't be. So it's very not important for you. That's really my question, because if you look at these things, they say,
I think language is the cleanest place to make the argument, but I'm not sure it's a unique place to make the argument. So if I knew reasoning as well as you did, maybe I could make the argument there, although I think there are reasons to think a lot of our reasoning isn't symbolic. Some of it is, and the symbolic parts of the reasoning that we do would probably be a place you could make the same argument. Someone who really knew motor control well, and especially planning, could probably make the same arguments from there. So the ways in which human beings can make very complicated plans where they're chaining together things they haven't done before, like how I'm going to get to his sound studio later, even though I've never actually been inside of it. There's a lot of abstraction, levels of different levels of representation about different events. Probably someone who knew that work well could make that argument. And Ray Jackendorf has tried to argue that there are deep analogies there. You know, whether it's one learning mechanism that allows us to acquire all of these complex bodies of knowledge, or whether maybe there's some domain-specific knowledge that's innate that goes into each of them, I don't really know. I don't think it's an accident that we can do this at many levels. I don't buy Spelke's claim that it's language per se that enables the others. I think it's more like machinery allows us to learn language. Language is a great mnemonic for acquiring the others, but that machinery is being used elsewhere. So compositionality is another way of talking about some of what we described. I think compositionality is a broad human property, and if I could make, if I knew the details of compositionality and motor control and planning, I might have been able to make the same arguments from there.
Okay, we got to stop. I got it right in there.
Now we had 2 hours and 15 minutes. We have to go.
Right, and the next talk starts. What time should the next start?
15 minutes ago, I think.
Half an hour. So the next talk is going to start in half an hour. Okay.
Was it really?